Burka, the Director of Histocompatibility and Immunogenetics Laboratory, University of Calgary. And our speaker today is a well-known person to every one of us, as Prof. Dr. Ilias Doxiadis. I will not talk anything about everyone, and I will give the floor to Dr. Burka to take it. So please mute yourself, and at the end of the presentation, you can also participate with your questions. All the best.
Thank you, Fadi. I am happy to be introducing Dr. Ilias Doxiadis. Ilias was born in Alexandria, Egypt. Is of a Greek nationality. He has received a PhD at the Institute of Genetics at the University of Munich. And he has done postdoc training in both at the Institute of Hematology and Immunology at the GSF in Munich and at the Department of Immunogenetics of the University Hospital in Essen in Germany. He headed the immunogenetic lab in biotests in Germany between 89 to 92. And later on managed he was the managing director of the Eurotransplant reference lab in Leiden, Netherlands, between 92 to 2013. After that, he was a consultant for transplantation and currently Transfusion Institute, University of Leipzig. He has been a long-term chair of the committee for proficiency testing at EFI, member of the executive committee of EFI and president of EFI. He also served as member of the tissue typing and organ procurement advisory committee of Eurotransplant. The list of achievement of Dr. Doxiadis is long, and I would like us to have some time to listen to an interesting topic entitled Analyzing Luminex Generated Data Using Machine Learning, AI. And without further ado, Ilias, please take it.
Thank you very much, really. I'm very happy to be able to present some results which we have generated the last 1.5 to 2 years. I will take you a little bit away from the classical diagnostic laboratory into, let's say, some new ideas which might help us, could help us in the future to understand a little bit what's happening with MHC. But let's start directly so that we know what's going on here. Yeah. Okay, the participants of this study, there's a lot. In Greece, the 3 major laboratories which participate in transplantation, in solid organ transplantation. In France, the Hôpital Saint-Louis in Paris. And the main person there is Ioannis Theodorou, to whom I have a very good contact. And we have built up the whole idea how to go further with the Luminex data. And of course, in Germany, together with Claudia Lehmann, which is head of the lab at the University Hospital in Leipzig, and her boss is Reinhard Henschler, who is, let's say, he's coming from the blood bank and hematology. Now, one paper is out, which is in Frontiers of Immunology, and one is now in the last preparations prior to send it away and then to be able to promote our work. What I will tell you today, and mostly some published data, but mostly yet unpublished data. So to give you an idea where we want to go also in the future and where perhaps we could have some collaboration within ASHRI. Come on. I don't know what. Okay. Now, when we talk about the MHC, we usually start to talk about the biological role of the MHC, which is on one side acts as a stimulator, inducing an immune response. You know very well when you do some MLCs, you need stimulators and then responders. Then also the MHC plays a role. So the non-self epitopic determinants of the stimulator will be seen as as foreign, and the body or the cells react. These are the two things which we know very well, but actually there is a third role of the MHC, which has nothing to do with the immune response and all this type of thing. But the MHC acts also as target of the immune reacting cells, or that means the T cells and the antibodies. So there are protein structures which can be attacked, recognized and attacked by antibodies and immune cells. So this idea came up after we talk a lot about it, and so this is my partner watching also the webinar. It doesn't like me. Okay, so what I would like to invite you today is take a helicopter view about this system MHC as a conglomerate of many proteins encoded or in the short arm of the chromosome 6, and which we would like to know in order to be able to predict future reactivities, what happens now. So, come on. So if we take HLA or MHC as a testing target, and our method is for sure the Luminex, because it is the state of the art how to do antibody screening. So we have on one side the class 2 determinants of the DRB1 locus. We have the second DRB genes, the 3, 4, 5, well, which determines, let's say, the ancestral lineage. We have DQA, DQB, DPA, DPB, all this for the class 2. On the other hand, we have A, B, and C, the HLA, class 1 determinants, E.g., FH, and we have also the MICA. We can type for everything in a very nice way, but also we can test in an antibody setting using Luminex against every one of these determinants. And this will give us the pattern a patient recognizes foreign antigens of the MHC. So the assay itself had also two different steps. Obviously, the detection of antibodies against the microsphere coupled targets, we know that, and the definition of the specificity. This is a trivial way what Luminex is giving you, but actually, the results coming out of the Luminex machine gives you information about the immune response of the patient towards the products of the MHC and defines patterns of the immune reactivity within the whole population you are analyzing. So this is a bit different of what we actually do in the diagnosis. We define antibodies, we define the specificity, and that's it. No, it's much more behind these two things, which we till now don't really evaluate. And we started with this idea. So I took this slide from the IMGT and I put some arrows onto it. First of all, where you have antibodies, DPB1, DPA1, you have against DQB1, DQA1, specific antibodies, the C, the B, and somewhere is the A here. We have antibodies against HLA-E, HG. Now, we ourselves don't have tested yet. G and A and F, we have seen the antibodies. A molecule complex here, DQA2 and DQB2, which indeed is expressed, but probably intracellularly. So we don't have a test kit for that, but perhaps in the future something will come out. We can type for all of these determinants, and we can screen for all of these determinants. So we have a wonderful system, which is very polymorphic. We have a lot of patients which we are already tested, and now we ask the question, what to do with this data? Do we throw them away, do the diagnosis, and that's it? Or can we continue further with the analysis? Now, we decided to go further with the analysis, knowing that working models of the epitope-based analysis, Duquesnoy, Birches, Eplerts, Tereps, and so on, which restrict their analysis, saying no immune response against self-epitopes are seen. I fully agree with that. if they are even if they are expressed by different loci. Like if you have some epitopes on the HLA-A and you have shared by HLA-B, you will not make antibodies also against HLA-B. We came up with something, no a priori assumptions, saying we are not using any cutoff at all, we are not deducing any NC, we are no other restrictions, we just put the data like we get them out of the machine and let the system work and see what's coming out. It was a little bit of funny when you are within the HLA system for so many years, there's many decades, and then you let the system work itself. So you cannot manipulate from outside, you cannot throw away data because you think they are not important, or the MFI values would be very low. Doesn't matter. The system will see its way through and will give you some answers, which, to my opinion, are not bad. Come on. So, what is a single bead assay? We all know. I will go very, very quick onto that. It is IgG specific. We have the Luminex beads, we have the antibodies we mix, you get some binding of secondary antibody after that, and you get an MFI value. Simple as that. It is a fluorescence assay which works in the majority of the cases, which gives you results in the majority of the cases, because we have also problems there in order how to define positivity and negativity. But for the system I will present to you today, we don't need that. We don't have an a priori positivity, a priori negativity. It's just the results which are coming out of the system. The bead itself, very simple actually. You have a dye impregnated bead with a color. Every single molecule has another color. You have the HLA molecule. We have the allo antibody, which is human in this respect. You have the secondary antibody, and then mix, wash, test. That's it. You have two lasers, and these two lasers give you, on one side, which bead you are looking and which antigen you are testing, and if it's positive, or to be honest, if it's bound the secondary antibody, yes or no. This is the question you have. You generate data which you would like to test further. So you have fluorescence values, which are quantitative variables of the sera, which are extracted from the files of the provider. Now, we all know fusion if you use one lambda, and the other one of Immucore, I don't know, because you are not using in the lab the Immucore kit, but I have seen results of the lab in Saloniki. They look similar to the ones of one lambda. Perhaps the values are a bit lower, but it's to our opinion, the only difference. The sera we tested are coming from pre-solid organ transplantation, heart, liver, kidney, or pancreas. Post-transplantation, we have hematological patients. That means patient expecting a stem cell transplantation, or platelet substitution patient, and so on. And also patients suffering from other diseases, for example, recurrent abortions or miscarriages, and so on and so on. So we put all the data together. Sure, analyzing first if they provide you similar results. So we didn't saw any statistical difference between solid organ transplantation patients, hematological patient on the other side. So we could pull the data and analyze them together. Per patient, we used here in these studies only a single serum. I am sure for future studies, we are going to include also many sera of a single patient, because there is a lot of information there which we would like to have at the end. The participating laboratories use similar experimental testing protocols and kits. All of them one lambda, and only one provided together with one lambda also Immucore data. So there were no real crucial difference of the labs. However, we analyzed the data separately. The Greek data together and the Leipzig data on the other side also together. So we didn't mix Greek and Leipzig data yet. We're going to do it in the near future, because I think it's a loss of information if we split the amount of data which we have. Now, what do we include in our analysis? Yes, for sure, the patient ID, the serum ID, the gender, date of birth, the identification of the lot, test date, the HLA type, the highest resolution possible. However, also because we started using Luminex quite a bit ago, so we use also some low resolution from the data. Perhaps it's not very clean, but on the other hand, it is an information which is a pity to lose. The fluorescence values of the antibody against the 100 beats for class 1 or the 100 beats for class 2 of both. And also we have also some information on the screening beats. Why to use the screening beats? Because we have there some additional loci which we can test, and this is the MIG-A. So also there, not to lose the information, we might need them in further analysis. So at the end, we have something, 150 items with about the number of patients to be tested. And here we have numbers from 800 to 3,000 tested patients, differing from the analysis we are doing and the cooperation we are having. We will start a new cooperation here with about 12,000 sera. So this will put our computers into the limit, but luckily we have the cloud which can help us in this respect. Now, problems. You have problems. It's not as easy as everybody thinks, because extracting data from a computer program is fine, but it's not easy because you have some missing or erroneous data. You have some names which are written a bit different, so you cannot couple patients to the old data. You have, in some respect, integrity of the data. You have a day-to-day testing which might influence your analysis. So you need a QC of your data. I mean, I'm coming from the QC business, so I know what it is, and this cost us, I have to admit, about half a year to clean the data. But when you are on this, let's say on this road, you must do it in order to have rather good analysis and to be happy in what you're doing. So we have in general something 200,000 to 5 million single values which will be analyzed or already have been analyzed, and we have two methods which we're now using. On one side, the Spearman correlation, and on the other hand, the PCA. We use those to visualize the results and perform the statistics. I will not give you any p-values. I will just show you what rolls out of the computer, because this is more impressive than having a p equals 0 point blah, 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 and so on. Data for class 1 are the patient population of Leipzig. The data for class 2 are from Greece, all the 3 major laboratories, and also the Leipzig data. So you will see what's happening if we analyze a cohort of Greece and you compare this cohort, or you analyze on the other side the cohort of Leipzig. We know that these two cohorts have different frequencies in terms of HLA antigens, but this doesn't matter because immune reactivity has nothing to do with the population frequency. It has only something to do of how a patient recognize, or a group of patients recognize, a foreign antigen. And this immunology is the same, to our opinion. Now, what is this Spearman correlation? One idea I can give you today, go please to Wikipedia. It is well written, and it is not only written for mathematicians, but also for, say, always dumb biologists, or let's say medical doctors, which they are not dumb. But it shows you what is the system. So intuitively, the Spearman correlation between two variables will be high when observations have a similar or identical rank. That means if they behave similar, then they will be, both of them, high and will be located on the same spot. This is a very simple idea behind the whole story. If an antigen is recognized by several patients in the same way, then the reactivity will always rank in the same position. Simple. And this, we made use of that analysis in order to take a look to our matrix. These are the simple shot of the matrix, of part of the matrix. You see here the HLA antigens, which has been coded on the beads, the negative control, the positive control, and then the different results which are rolling out of the machine. Now, Spearman's correlation, the first ones has been performed in psychology. When you think psychology and modern statistics has nothing to do very much, but it has. So the idea is very simple. If the sign correlation indicates the direction of association between X, this one, the independent variable, and the Y, the dependent variable. So if, come on, this one. So if both of them increases when the other one increases, you get a positive correlation. If the one increases and the other one decreases, then you have a negative correlation. Simple. Look at please at the Wikipedia, then you get more information on that. Now, why we would like to compute a correlation matrix? First of all, we have a large amount of data, which our brain cannot see immediately. Even with a history of 50, 60 years of HLA and the knowledge which we have in the laboratory, it's very, very difficult to summarize the large amount of data. And it's difficult also to visualize possible patterns. And this we wanted to have. And then to input these results into other further analysis. Why? Because we want to predict reactivity of a new coming patient. At the end, it is also a diagnostic tool when checking other analysis. You want to know if a patient reacts against this bunch of antigens, it might also react with another bunch. This is very important. The output, positive correlation displayed in blue, and I will show you the slides, and negative correlations in red. First of all, we don't have any negative correlation at all in our database. It will not happen because we always have positive correlation to date. The color intensity, also nice to know, is proportional to the correlation coefficients. So reordering of the antigens goes according to the correlation coefficient, and then we try to identify hidden structures and patterns in the matrix. Well, it's nice to have, but it's also give us information of how a patient reacts against some of the HLA antigens. What we see at the end is not the whole and the full story. It's impossible. We see just the tip of the iceberg. And what we would like to do with time is to put this tip of the iceberg to make it as large as possible so that we can really say something about what happened beneath here. Let's see what the future will tell us. For class 1, this is what's rolling out of the computer when you feed it with the data of class 1. So you get the so-called correlogram. This is always half of the whole story. I will show you the whole on the other side in a couple of minutes for class 2. Here you have the antigens ordered by their coefficient to each other. This is the same number of antigens. We have, and the near the antigen are to each other, the higher or the better are recognized by the giving serum or the giving sera, to be honest. Now, a zoom into it is not really very professionally made it because my computer don't like this type of things. I have to use it in a bigger computer. I don't have so much memory. But you see some things which are very nice. For example, the B12, we know, 44, 45. We know also that 45 actually is a BW6, The 44 is a BW4 in the BW4 group. And we have this little B1512, which is within this group of antigens. Then we have the BW4 group. And here we have also some peculiarities. For example, the 24s, which are within the BW4, are located in another position than the 25 and the 32. Although all of those A locus specificities have the BW4 antigen. That means, if we see it like this, that they other epitopes which are carried by 25 and 32 influences their position than the BW4 one. So this is the interpretation which we gave. Then we see also the group here with stars, which are the members of the so-called R complex. We have the R complex within the BW4, this 52, 51, and 53. And they are not, let's say, going together with B35 and B18, which are the other members. Here's the B35, B18 is further down. That means the BW4 in this respect is not as strong as the, otherwise the R complex is not, as a crag, is not as strong as the BW4 epitope. So if you go through the whole matrix in this way of thinking, then you see differences in the antigens of the HLA. That means if we want to have a very clear picture, we need many more data than the ones which we have here. And this is approximately about 1,000 patients which we have analyzed. Now, the BW6 one, the two stars are the 35 and the B18, which are on totally different position than the BW4 upstairs. We have here the group of the B13 with 2705 and B47. All serologists will say immediately, well, I know this is standard. When you have an antibody, then you react also against all these specificities. And then we have the B7 crag down here. This is the B7 molecule. And then you have the 60s and the 48. And also here, let's say the B22s and so on and so on. So with a simple click, 1,000 data, you have the history of HLA in front of you. This fascinated me the first time when we saw this picture. So what we have done in so many workshops, in so many analyses, with the modern techniques, and especially the computing techniques, we can do now with a click. And that means it offers us much better possibilities to go further. And one of those, I'll show you here. HLA-C, this is one group of C antigens, this is the other group of C antigens, and there is a difference between the two. I'll come in a second on that. That means you have distinct subgroups within the different HLA-C determinants. They have nothing to do with the microRNAs which have been published by Mary Carrington's group. They're something peculiar only for HLA-C and nothing with additional expression or non-expression. It seems the expression level doesn't play a big role in this analysis. The HLA-A2, sure, you know, serologists, A201, 69, 68, and then you have the group here, which I say more or less the 19 of the rest of the HLA, if you take the A3, A11, and so on outside. You have the 34, which we know is many times associated in the sera with A28. We have here the A10, here the 29s with 74, also very well known, the remaining of the 19, and then the standard A1, A3, A11. Now, HLA-C, I promise you I will come back. This is the picture a bit bigger. We have here the ones, the HLA-Cs, which are associated with the NK2 reactivity. They have an S in position, a serine position 77, which is nothing else than the BW6 epitope. So we have the Cs with the BW6. We have the other Cs here, which are associated with NK1 cells with an N in position 77. If you go back to your lab and you have specific BW6 sera, you do the Luminex, and then you get, depending of the specificity of the serum, you get this BW6 positivity and BW6 negativity. Now, I can show it also in terms of polymorphic residues. These are the one with the serine here, which is the BW6, and this is the NK1 with the N on position 77. Now you can merge and play around, and then you can see other groups. That's for sure what you expect, the C3. Then you have the 7, which is nearby the 0102 and the 1402. And then you have the darker you have is the better correlate the sera are. We know from the serology we have some ideas about what's going on there. I was fascinated to see how easy we can get results. And when we included about 3 times so many sera into it, we saw clearer pictures, and all these blurry surfaces here will disappear. This will come in the near future when we analyze this 5,000 and so on sera of our colleagues in Berlin. Now, leaving class 1, going to class 2, it is a double pictures, more or less. Until now, I showed you only the half of it, but you can see also now the whole picture. It doesn't change because it's like a mirror here. The information which we have here additionally is the degree of the, let's say, correlation coefficient, for example, between DRB1 1503 and DRB1 1601. So it is not 100%, but is in the majority of the cases, it gives you this pie with the different analysis, different numbers into it. Prima vista, you see the DR2s here with 15 and 16. You have here the, let's say, the DR9s. Then you have, funny enough, the DR1s and DRB5, the DR4s and the DR52 related. Important for our manner to look at the story is 1401 and 54, the 1454, which are virtually identical. We know that. We know that by sequence. We know where the mismatch between the two is outside the region where the antibody can bind. But this comes on the same position and raise the question, do we indeed need a beat for every single new allele which is observed in a population? Or can we deduct the results because of similar antibody reactivity? I don't like to have 500 or 600 or 2,000 different single reactivities in my vial and to analyze them after that if they are the same. So it will help diagnostics if we can establish a matrix and say, fine, 1101, 1104, for example, here, behave the same. So we don't need 1101 beat and 1104 beat. This is also like the 1401 and 1454. We will see if the companies and especially if the societies will accept this type of things, or the customers. For DQ, we have the DQ3s, we have the DQ4, you have the DQ2, and you have the DQ1. As we expected from the history of HLA. That means we are not on a bad side, we're on a good side, and we can now ask questions which we will never be able to ask this type of questions before. I will come to that in a moment. DP is fascinating because I have to admit, I have nothing to do with DP. It was at that time only by cellular manners. now we have serological means through the single antigens, and now we can test, for example, behavior. What's happening if I have a DPA different, but the DPB is the same? I'll show you in a second a slide on that. It's funny, it is different. That means sometimes we have to think about where the immune reactivity goes in this respect. Goes against the DPA or against the DPB. And this might help us in the future how to understand DP, not from the cellular point of view and permissive and not permissive, but from the serological point of view in terms of antibodies. DRB, very nice. And these are the Leipzig data. We see here the DRB1, the 4s and the 5, and the 401 and the 404. It's mimicking our knowledge with an additional information. Here again, the 1401 and 1454. 1303 together with 1301. So the difference between these two is not in terms of DR, but more or less in terms of the haplotype, which is then the DQ1 versus the DQ7. You can go 1101, 1104, identical behavior, and so on and so on. It is really sitting, looking at this, and yeah, and let your brain walk through all these spots and give you ideas, at least for me. DQA and DQB, similar story. Here we have the ones, here you have the twos, and then we end up here with a conglomerate of the threes. Always following the same rule. No having any rule, let the machine give you the results. And I think this is fascinating. The machine makes exactly the same, or in most of the cases, of what your years of serology would have done. I think we are really in the good track. DPA and DPB, I'll leave it like this to you, because I would like to go to the second set of analysis, which are the PCA, which is the principal component analysis. It is the most ancient and established technique for dimension reduction. It is simplest through eigenvector-based multivariate analysis. Fantastic. I have to admit, it took me a couple of hours and days, and perhaps weeks, to understand all this eigenvector-based multivariate analysis. However, it is a simplification of a complex reactivity. It has been invented almost 120, 130 years ago by Carl Persson, and then developed by Harold Hoteling. Wikipedia is a very good source for this information to understand what's going on then. It is mostly used as a tool in exploratory data analysis and also to make predictive models. That means this is why we need this type of modeling in order to be able to predict, let's say, our reactivities. And now it's to the point that we have a population analysis here. And from this population analysis, we would like to go back to the patient-specific analysis. And this we need, for the future, additional softwares and ideas how to do it. They exist. The problem is you need a very strong computer to be able to analyze that. So for the PCA, we can visualize genetic distances. We can relate different population, genetic distances back, and have a pattern recognition on that. It is rather simple to do it in this modern way because you have the programs, you click the button, and if your data are clean, then you have your results in a good way. This is class 1, very complicated. I mean, when we saw it the first time, we said, oh boy, I don't see anything. True. This is 2 dimensions. You have 1 dimension here. Oops, wait a minute. Here on the upper side, and the 2nd dimension here on the lower side. That means these antigenic groups here are different, totally different than the ones here. And when you look closer, you will see, yes, I can accept. Here you have more or less the A's, and you have more or less the B's, and they are different in terms of antigenic determinants. Now, you can add a 3rd dimension. Wait a minute. No, I'll go to the C first, prior to go further, because it's a bit clearer. For the C's, we have the same story. Here you have a group of antigens, and here you have the other group of antigens. So, and these are, if you have a reactivity of a group of patients against CW5 or C5, they will not react against C3. So these are really dissected into 2, but you can add a 3rd dimension into the story. And then you have here the C3 totally independent from the other C loci. Here, for example, you have the 7s, you have the 12, the 16s, the 14th, and these are different than the C3. So this gives you an idea of how a group of patients react against a specific determinant. And this is what answers you the question about immune response against these determinants. It's not a patient who reacts against that, it's a group of sera, a group of patients. Now, for class 2, again, very, very complicated to see because of also 2 proteins forming one, let's say, forming 1 DQ molecule. We have, when we add the other dimension into it, then the story comes a bit clearer. These are the Greek data generated from 1,700 individuals. Then you see the DQs cluster together, the DRs cluster together, and the DPs cluster here also together. Different. These are different antigenic determinants recognized by a whole bunch of sera. Now the idea is, do I have a difference between DPA and DPB? Yes, indeed you have. You can fix, for example, the reactivity of the sera towards a single DPA molecule. And then you ask the question, do I see differences in my population? Indeed, you see that. Here, the DPA 103 is fixed, and here you have the other DPs. A group is here, and the other group is there. So between these, there is, in terms of DP, a big difference in terms of reactivity against DP. You can go to the further dimension, and this makes the story much nicer. Here you have one group which is totally different than this opposite one, and here one group which is totally different than this one. This type of experiments at home or in the laboratory, in the computer, allows to understand on one side the structure of the molecules, but much better the reactivity of the patients as a population against these antigenic groups or epitopic determinants. You would say, do I need that? To my opinion, you need that because you would like, in a step further, to predict. You have a patient with a specific HLA type, and then you want to predict which will be the antigenic determinant which the patient will produce, let's say, antibodies, and how big will be the chance that this patient will do that. So I can tell you we are in a very simplistic way trying to define that, And I was astonished that indeed it is possible, but you need much, much more data than the one we have here. Total of class 2, a mass, is not the mass of the picture. The mass is when you put to every arrow the respective name. This is a problem of this story. Again, second dimension, or the additional dimension, you have the DRBs together, the DQAs together, the DPBs together. What you would expect from, let's say, nature of the molecules. If you leave, if you go to a rather smaller population, like the one in Leipzig, we see here again, here the DR1s, DR9, DR10s, which cluster together, and here we have the 14s and so on, which are clustering also together. 12, I mean the DR52 associated ones. If you took the additional dimension, the situation will be much more clearer. You see the DR4s clustering in one position, but funny enough, the DRB4s also clustering together. These are the 9, these are the ones, and so on and so on. So it has now at least information which we didn't expect it from the very beginning to have with a click this story, but we have to go now further and ask a very specific question to our system. We can do that because we have the data in a computer manner. And I hope that this will give us much more information in the near future. The DQs, the same thing. The DQ1s, 5, 6s. Then you have here some 4s and the 3s, so, and the 1s. So they cluster in a way which you would expect. And you can use this for your further analysis, especially asking the question, what's happening if I put on the other hand the HLA type of the responder? What is the cluster which will pop out? That means if you have now, for example, a DR13, DRB1 1301 and so on the side of the responder, and ask the question, which will be the antigens this individual will recognize? This individual will recognize very, very specific molecules. If you give them, they will make antibodies against it. And this is our prediction way, which we are going for the future. Perhaps we're going to ask to be part of a workshop. I don't know yet if we have the means to do that, but this is the idea which is behind the whole story. Now, I think I bothered you a lot with all these arrows and the correlograms being DRBs or being DQBs or being DPs, first and second, third dimension. I would like to actually to stop now the whole story by summarizing and concluding some things. First of all, we are on a good track that machine learning or artificial intelligence allow us to understand the complex data in histocompatibility. And when I say the complex data in histocompatibility is not only antibody production, it's also the immune setup of the respective individual. Because then this is triggering more or less the immune response against the foreign, and at the end will give us the idea what's happening there. Now the 60 years of HLA history could be repeated here with just a click, and 800 to 3000 sera is not really a lot. If you have the programs, you do it really with a press button. And I think the most important is including additional data from many, many laboratories will allow us to dissect the immune response to the factors of the HLA and predict patient-specific responses towards the mismatches or the incompatibilities. Again, thank to all the people who have done work here. And I have to mention again Yanis Theodorou, who was the one who made the analysis in Paris, and which we had a lot of discussions by telephone, and we have been there for a couple of times. And then our group here in Leipzig with Claudia Lehmann and myself. This paper is out, and I hope when I have the next one, I'll send it to Ashley. So thank you very much for listening to me, and I hope that you're not totally confused, but for sure you are confused. Thank you very much.
Thank you very much, Dr. Doxiadis. At this time, we'll open the floor for questions and comments. So if you're comfortable to speak directly, just raise your hand. And if you want to write your comment on the chat, you are also welcome to do that. I have somebody under the name of Archie asking to speak. Who's Archie?
It's me.
All right, Freddy, go ahead. You are
yeah, it's me. Thank you, Dr. Ilias, for this wonderful talk. I do have actually two questions, if you allow me. Number one is, are you in agreement with scientists who state we can see antibodies specific to DQ alpha chain, not DQ alpha beta as combination?
Oh yes. At least when we perform the analysis and we restrict, oops, I don't know, I have something black here in the middle, and we select data looking only for DQ alpha and then ask if we have a co-reactivity with DQ beta, we see anti-DQ alpha specific antibodies, yes. Okay. If I got your question correctly.
Yeah. The second question is, you state at one of your slides that because of this correlation and statistical agreement study that you show us here today, we don't need to have much variety of alleles with the same antigens in every bead. However, clinically, we do still see allele-specific antibody that shows in a certain circumstances positive cross-match versus negative cross-match with absence of that antibody. So how you can link your statistical finding versus what we see clinically?
Okay, what the statistic shows you is with a bunch of patients, you will have these results. If you go to the singular patient, it's another level, you will see allele-specific antibodies. I mean, we start with the very simple ones, anti-A205, anti-A201. You have patients which are A205 and produce antibodies against A201. This patient have the mostly of the times a B50, CW6, DR7 haplotype. This is known for many years. We know the 6601, 6602, but this is another story. I say here that, for example, B1401 and B1454, they have the mismatch somewhere in the transmembrane or one of the other exons, which is not influenced, or at least is not recognized by antibody specificity, by an antibody. The same holds true with 1101, 1104. These are two alleles which always will be seen with the same antibody. So if you have an antibody against 1101, this antibody will recognize 1104. I mean, in this respect, I say, fine, if we do our analysis correctly, then we can say the bead 1404 and use another bit instead of that. This is something which you should discuss. I fully agree with what you say with allele-specific antibodies, but we have first step by step to go further and say, we see it, what's happening in the situation in the clinic. Do we see the differences? And then we can continue. We will not do it to say, forget half of those and then it will be cheaper. No, that's not our way to do. We have to test for that first. Thank you.
I have a question here, Ilias, from Barty Mittel, kind of a comment. He said that he believed that every country should generate such data for prediction. What's your opinion?
If they have enough data, yes. I would suggest, though, that as a first step, we do it for all of us together. We pull the data. We have to test, first of all, if we can pull them. Then we pull them, then we make general rules. And then we, from this data set, since we have quoted which data are coming from which country, then you can do the same story country by country, laboratory by laboratory. But we need some general ideas first, prior to go, everyone is cooking his own soup. I'm afraid that you will not have so many data. We use all the same kit. At the end of the story, it is the same kit, the different machines perhaps, but it's also more or less the same machines. And you expect more or less the same data.
Very good. Question from Dr. Ahmed Mustafa from Saskatoon, Canada. How can we correlate this work to epitope analysis? Both these tools can help us to predict antibody formation against HLA.
Shukran. This again something which I'm very keen. We saw if we include many data. I couldn't show all the slides which we have generated, but we see within groups, for example, the C locus group, two major groups, but there are within these groups, you see some little pyramids. And these are, funny enough, correlated with epitopes, which has been already described by others. And this is the next step in the whole story. I think what we see with this type of analysis is more or less immunogenic epitopes are popping up and the others are hidden behind it. I tried to explain it with the B12, 44, and 45. I tried to explain it with the R complex, BW4 ones, so that you see the epitope, the immunogenic epitopes are pushing an antigen in another direction than what you would expect theoretically. This is, I think this analysis will help us, but we need large amount of data, in a half a year perhaps. Oh, so.
Okay. I have a comment here from Hanouf Zuhair. Thank you, Dr. Doxiadis, for this interesting talk. Mine isn't a question, but a comment. If all this data is generated by your overwhelmed computer, can't wait to see the data analyzed by a supercomputer.
Okay, you cannot do it with your laptop. You cannot do it with the computer at home. You have to use a cloud. That's the first thing. For sure, if we have, let's say, 50, 60, 70,000 sets of data, then we need a rather big computer, strong computer, to be able to analyze those. We'll be able, it will happen, but step by step. Mm-hmm. And if you have access to supercomputer, please inform me and we can do something together.
Very good. While waiting for other people to write their comment or ask to speak directly, you made a comment that it doesn't matter the level of expression when you were looking at the first algorithm.
Yes.
Can you expand on that a little bit?
Yes. I mean, you know the Mary Carrington microRNA story, that some of the HLA-C alleles are not so strongly expressed or are not expressed on the cell surface because of this microRNA. Now, we all know that HLA-C has a differential expression on the cell surface. If you take platelets and you use a monoclonal antibody on the other side, for example, CW1, and you take 10 individuals, you will have 10 different reactivity patterns. Some of them typed for CW1, you virtually see nothing on the cell surface, and others you really have a very strong response, equally HLA-A and B. So we know that. But this does not seems to affect so much the data which we got or which we get from the Luminex, because we see even with low MFI values, we see positivity. And this is the good thing of the whole analysis which we have done here. We don't take a cutoff into our analysis. We leave it totally out because we see the patterns, even with low NFI values. This will be for some of the C locus, for example, a bit higher than other values. And this is enough for the machine learning and the correlograms to give you positive results. This is the fascinating story. You don't need to have very, very high values in order to see something. You see them also with low values.
I mean,
because of the correlation.
Yeah. In the clinical practice, we've seen that MF5 of up to 5,000 in CW DSS do not really clinically matter.
Correct. Correct. I fully agree with you in some respect. Mm-hmm. But for other patients, for example, if you look longitudinally to a patient after transplantation and you see rejection episodes, you will see a delta value of, or let's say an MFI value for 3, 4, 500 is correlated with a humoral rejection. You can tell, okay, all the antibody is in the kidney, and therefore I don't see it in the periphery. No, but you have also HLA-A2, for example, 20,000, 22,000, but for C locus, low values. I fully agree with it. Therefore, we decided not to use cutoff values for this analysis. But naming cutoff, I don't know if you're familiar with the data of Peter Stastny, how he analyzed Luminex data, where he said a cutoff value of whatever for all the beats to be the same is wrong. Something is behind this idea of Peter. He is correct. We should not use a cutoff value for all beats, but we should try to define individual ones. A beat cutoff, yes. I will not show you data here today, but the difference is strong, significant from one beat to another. Mm-hmm. But I think this is something which we can get out of a cooperation with a lot of data.
Sounds good. Ashraf Dadda from Jeddah, would you like to speak, or I see that you're writing a question, a comment. Go ahead and speak.
Yes, thank you very much, Dr. Nuruddin, and thank you also, Dr. Ilias, for this presentation. Unfortunately, I have just joined the meeting because I have urgent corona cases in the hospital. Oh, so I was not able to join from the beginning. And I would like to ask you, Ilias, if you find the role of the artificial intelligence in the routine praxis, do you think that will be find its way to be established in a small and middle facility, or will be limited to large ones or universities?
Okay, let's say if you have a big facility, you can generate a lot of data in a very short time. But if you have a small facility, it's very difficult. But on the other side, you can profit of the data generated elsewhere. And that means you need good cooperation. We have, for example, a very good clinic on leukemia diagnostic in Munich, where they managed to put together all the possible laboratories in Munich in order to generate the data. And they can have diagnostic in very, very short time. Patient comes in a couple of hours, so let's say one day later, you have a full, precise diagnostic, also with information regarding survival, and so on, and where do the patient will go after that. This can only be done in very, very big labs. On the other hand, if you are able in your clinic to have all the information of the patient in such a way that they are computerized, then with simple programs, not today, but less than a couple of years, you can have a much, much better diagnostic than what you have today. And that means artificial intelligence will go also into the middle sized laboratories, and from there also the small sized laboratories, like it happens also with the methods. At the beginning, we had RFLP for DNA typing only to be done in rather big labs. Now we have NGS, and NGS also middle-sized labs can do it now. And I say the lab which we have in Leipzig is a more or less middle-sized laboratory, but NGS is you cannot do without it at present. And that means in a couple of years, this will go also to smaller labs because it's cheaper than every other typing method you can apply in your surroundings. So in a way, yes, I expect AI to evade, let's say, the laboratory because the knowledge is there. That's it, to my opinion.
Thank you.
Welcome.
We have a comment question here. What could this result have clinical relevance? And for example, could we consider this plot for virtual cross-match?
Yes.
Nice question.
Yeah, it's a nice question. Yeah. Yes. I think, or let's say, I'm sure that this data will allow you to better understand also a virtual cross-matching than what we have now. We do a screening and then that's it, and you put it, and then we look at the data, that's fine. This is the unacceptable antigens, and therefore we don't want them, and this is a virtual cross-match. But the more you go into the detail in this type of analysis, you have other insights. And I think there we will profit from this type of analysis for the future. To predict, yeah. And I think this is at least my goal. Always wanted to have that. I have a dream, let's say it that way.
Excellent. Last chance for a comment or a question. Many people are thanking you for this exciting and stimulating presentation. Thank you very much, Dr. Doxiadis. I think you are giving us food for thought here. And I'm sure that the Archie community will stimulate some more discussion on this topic. So thank you everybody for attending, and I'd like to thank your partner for giving you the emotional support right there in the room.
Thank you. Yeah.
All right. Thank you everybody. Have a good day.
Thank you.
Well explained, Dr. Barka. Well explained.
Take care.
Thank you very much, Ilias. Thank you, everyone. I appreciate
Welcome.
it.
Thank you for giving me the opportunity to talk. And looking forward, perhaps we should organize a cooperation.
Absolutely.
Okay. I wish you a wonderful evening.
Thank you very much.
And,
And thanks to your partner too.
Yeah, you have your weekends. We need a couple of days longer, but that was a nice thing in Egypt when I was at the last year of school. We had the Friday free, and then we had the Saturday free, and we had the Sunday free. So it was a 4-day school. That was good. Not long enough, but it was. So I wish everybody nice evening, enjoy, and we surely will talk in short time.
All right.
Okay.
Stay
Thank you, yes.
safe, everybody.
Schönen Abend noch.